Hello, this is Dr. Eric Bremer, the Chief Scientific Officer of Precision Biomarker Resources. In this presentation, I'd like to discuss the importance of microarray data quality assessment as a key step in obtaining interpretable and hopefully biologically meaningful results from your gene expression or microarray experiment. And I'd also like to walk you through the quality assessment report that we provide to PBR customers for their gene expression experiments. As in any experiment, understanding the variance is essential to results interpretation. Even with the technological and protocol improvements that have made microarrays analysis a robust technique, it's still a very complex technique when variance and noise can come from many sources, including technical and even biological sources. Examples of technical causes may be the platform employed, the RNA extraction procedures employed. There's amplification, labeling, hybridization experiments that steps that need to be controlled for. In addition, there are biological causes of variation that may not be what you're actually looking for. They can come from the tissue itself, tissue contamination. This is particularly a problem in tumors analysis, is that sometimes they're mixed tissues. There can be clinical covariants such as age, sex, race, etc., and cell cycle differences that can all influence your gene expression experiment. Ultimately, the goal of quality assessment is to identify and minimize the influence of these covariates. So the question in this slide is really, when is it important to do this quality assessment? After generation of the cell files, data is typically imported into a data management system, and then the files go through some sort of series of pre-processing steps, such as background correction, normalization, prior to detection of differentially expressed genes. And ultimately, you probably want to do some sort of higher level analysis, such as gene set enrichment or pathway analysis. At PBR, we like to do quality assessment prior to and after the pre-processing steps. Prior to or on the raw data helps to give a first look at the overall chip integrity and the RNA quality that went into the microarray. And it may also indicate the type of normalization or scaling that is most appropriate or required for the pre-processing steps. After normalization, it provides information on how appropriate was the normalization process. It can also identify chips that are still outliers. If outliers are still present, one might want to remove these outliers and redo the pre-processing of the chips before going on with further analysis. Later in this presentation, I'll provide an example of how removal of outliers after careful quality assessment can greatly improve your results. The quality assessment we provide at PBR is based on the bioconductor package Array Quality Metrics and implemented in R. This package looks at metrics from each individual chip and also provides some nice comparisons to compare between chips. When we look at each individual chips, look at intensity-dependent sort of ratios, such as M versus A plots, and actually look at spatial or effects or defects in the chip by just kind of image analysis of the chips. It's also important to look between chips because you're really concerned about the homogeneity of fluorescence intensity, and this is where one can detect potential outliers. And it also may give you a first clue as to biological meaning, and I'll explain this as we go through an example. So let's now look at an example of a QC report, and we'll go through each of these comparisons one by one. To provide this example, I've chosen an experiment that's publicly available on Array Express. And in this is looking at mouse gene expression, looking at 5 wild-type mice versus 5 knockout mice of this gene ATNX1. And this is actually on an older generation chip, mouse Affymetrix gene chips. But there's some good reasons for looking at this experiment. The first thing that's provided with our Precision Biomarker Resources quality assessment report is kind of a summary. And this summary provides the name of the array when it was scanned and the different tests that were provided. It's an easy way if you just want to see how well your chips performed. This is a good place to start. Every place you see an asterisk suggests a potential outlier array, and in each column which test suggested that it might be an outlier. Now, it's not surprising in unscaled, unnormalized, or raw data to see several sort of asterisks in your summary report. The first plot then you see in the quality assessment report is the so-called M versus A plot. Now, this plot creates a theoretical chip that gives the mean intensity for each gene across all and ranks them by intensity, each spot across all chips. What's helpful and then compares each individual chip to that. So if we look in the upper left and where it says number 1, the blue provides where most of the density is, and it's going off the center line, off of 0, indicating that this chip may have some issues with background or saturation in the upper end, because at the higher intensity ones, or to the right of panel number 1, it's there's a drop off. If we look at the next one, number 2, you can see the highest density, the blue line, is about on the 0 line, suggesting that this is probably a reasonable quality chip. The next plot is simply looking at all of the pixelated intensities. This can give a good idea if there's a defect in the chip or if there's some area that needs to be addressed. In this case, we can see there's no obvious sort of defect or spots on these chips. Now we want to start looking at the distributions of intensities, because brighter chips, if they're biased in one group versus the other, may just actually give you large false discovery type of rate. One can look at this as either in a box plot or a histogram. Histograms plots simply the density or the number of spots on the chip that have a certain fluorescence intensity, and you want these to be as close as possible. The second thing is looking at the box plots. The line within the box indicates the median fluorescence intensity. The box, where most of them are blue, indicates the middle 2 quartiles of fluorescence intensity, and the whiskers, or what look like error bars, provide the range of fluorescence intensities. What we've done here is 2 of the chips with the gray boxes are highlighted in gray as opposed to blue because they appear to significantly deviate from the rest. Also, I said it's important to look between chips to see what is how even their distribution is. In the heat map, which is kind of a hierarchical cluster trying to show how close or far apart these chips are, we can see 2 chips over on the left, number 6 and number 8, that have a yellow intensity indicating that the distance the overall distance between them and the other chips is significant. These were the ones that we saw in the box plot that were gray and had a lower overall fluorescence intensity. Another way of looking at this is principal components analysis. Principal components analysis drives multidimensional data down to a few dimensions and tries to create a vector of which is the highest degree of separation. In this case, we can see two chips over on the left-hand side that appear to be separated from the rest of them, and these also turn out to be the same two, number 6 and number 8.